Hello friends and welcome. In today's deliberation, we shall talk about strategies in the search for new lead compounds. To begin with, let me give you a brief introduction about the topic. The primary objective of medicinal chemistry is the design and discovery of new compounds that are suitable for use as drugs. The discovery of new drugs not only requires its design and synthesis, but also the development of testing methods and procedures, which are needed to establish how a substance operates in the body and its suitability for use as drug. Now, one might ask, what is a drug? Drugs are defined as chemical substances that are used to prevent or cure diseases in humans, animals, and plants. That was a brief introduction. Now, let's shift our focus to the main objectives of today's topic that will help us to know what is a hit and a lead and how they help in discovery of new drugs, to understand the strategies used in discovery of new lead compounds, and also to understand these strategies and how these approaches possibly direct to synthesis of new drugs. Let's first of all try to understand what is a hit and what is a lead. A hit is an active substance having a preferential activity for the target and which satisfies certain criteria, that are reproducible activity in a relevant bioassay, confirmed structure and high purity, specificity for the target under study, confirmed potential for novelty, and a chemically tractable structure, that is, molecules presenting a certain affinity for a target. Once a hit is discovered, its activity must be confirmed and validated. Typical hit validation criteria are as follows. The hit must be active in vitro and be amenable to in vivo activity in target or disease models. The hit should not display human ether agogo related, that is HERG, toxicity. The analogues of the hit must display clear structure-activity relationships, that is SAR. Basic physicochemical and ADME properties of the hit series must be evaluated in order to identify potential unwanted properties in the series and to assess structure properties relationships, SPR. The hit should not contain chemically reactive functions, and the hit must provide patent opportunities. Only then does it become a lead substance commonly named lead. This topic deals with the strategies that have been used in search of the new lead compounds. A retrospective analysis of the ways leading to discovery of new drugs suggests that 5 successful strategies can yield new hits and/or lead compounds. First strategy, analog design. Second strategy, systematic screening. Third strategy, exploitation of biological information. Fourth strategy, planned research and rational approaches. Fifth strategy, applying biophysical technologies and computational methods. These consist of the improvement of already existing drugs, of systematic screening, of retroactive exploitation of biological information, of attempts towards rational design, and of the use of the target protein structural information. Let's discuss the first strategy, that is analog design. The most popular strategy in drug design is the synthesis of analogues of existing active molecules. The objective is to start with known active principles or first-in-class drugs and by various chemical transformations. Prepare new molecules, sometimes referred to as follow-on or me-too compounds, for which an increase in potency, a better specific activity profile, improved safety, or a better formulation that is easier to handle by physicians and nurses or more acceptable to the patient are claimed. A typical illustration of this approach is found in the series of losartan analogues or in the conazole series. All the compounds show similar structures and similar affinity for the angiotensin II receptor. As such, they can be considered full analogues. Different categories of analogues. This definition implies that the analogue of an existing drug molecule shares chemical and therapeutical similarities with the original compound. This definition suggests three categories of drug analogues: analogues presenting chemical and pharmacological similarity, analogues presenting only chemical similarity, and analogues displaying similar pharmacological properties but presenting totally different chemical structures. Analogues of the first category, presenting at the same time chemical and pharmacological similarities, can be considered as full or true analogues. These analogues correspond to the class of drugs often referred to as Me Too compounds. Usually, they are improved versions of a pioneer drug over which they present a pharmacological, pharmacodynamic, or biopharmaceutical advantage. Some examples are the angiotensin converting enzyme ACE, inhibitors derived from captopril, the histamine H2 antagonists derived from cimetidine, and the hydroxymethyl glutaryl coenzyme A reductase inhibitors derived from mevinolin. The second class of analogues, which are made of chemically similar molecules and for which we propose the term structural analogues, contain compounds originally prepared as close and patentable analogues of a novel lead, but for which the biological assays reveal totally unexpected pharmacological properties. A historical example of the emergence of a new activity is provided by the discovery of the antidepressant properties of imipramine, which was originally designed as an analogue of the potent neuroleptic drug chlorpromazine. For the third class of analogous compounds, chemical similarity is not observed, but they share common biological properties. We propose the term functional analogues for such compounds. Examples are the neuroleptics chlorpromazine and haloperidol, or the tranquilizers diazepam and zopiclone. Let's come to the next strategy, that is systematic screening. This method consists of screening new molecules, whether they are synthetic or of natural origin, on an animal model or any biological test without having in mind hypotheses about its pharmacological or therapeutic potential. The trend is to undertake in vitro rather than in vivo tests, binding assays, enzyme inhibition measurements, activity on isolated organs or cell cultures, etc. There are many types of screening methods which I am explaining. The first one is called extensive screening, which is generally applied to totally new chemical entities coming from an original effort of chemical research or from a laborious extraction from a natural source. For such molecules, the high investment in synthetic or extractive chemistry justifies an extensive pharmacological study, central nervous, cardiovascular, pulmonary and digestive systems, antiviral, antibacterial or chemotherapeutic properties, etc., to detect if there exists an interesting potential linked to these new structures. In summary, a limited number of molecules are studied in a thorough manner, that is, vertical screening. The extensive screening approach has often led to original molecules. More recent examples of successful systematic screening campaigns are seen with the discovery of cyclopyrrolones, example zopiclone, as ligands for the central benzodiazepine receptor, or of Taxol as an original and potent anti-cancer drug. Then there is random screening. In this case, the therapeutic objective is fixed in advance, and contrary to the preceding case, a great number of molecules are tested, but only on a limited number of experimental models. This method has been used for the discovery of new antibiotics. Among more recent successes of this approach, one can mention the discovery of lovastatin, also called mevinolin, which was the basis of a new generation of hypocholesterolemic agents acting by inhibition of HMG coenzyme A reductase. There is another type of screening which is called high throughput screening, or HTS. The arrival of the robotics in the 1980s, along with the miniaturization of the in vitro testing methods, meant it became possible to combine the two preceding approaches. In other words, screen millions of compounds on a large number of biological targets. This strategy has delivered numerous drugs. HTS is usually applied to the displacement of radioligands and to the inhibition of enzymes. The present trend is to replace radioligand-based assays with fluorescence-based measurements. Another type is screening of synthesis intermediates. As synthesis intermediates are chemically connected to the final products, and as they often present some common groupings with them, it is unlikely that they equally share some pharmacological properties. Among drugs discovered this way, one finds the tuberculostatic semicarbazones. They were initially used in the synthesis of antibacterial sulfathiazoles. Subsequent testing of isonicotinic acid hydrazide destined for the synthesis of a particular thiosemicarbazone revealed the powerful tuberculostatic activity of the precursor that has become since then a major anti-tubercular drug, isoniazid. Another type is new leads from old drugs. Selective optimization of side activities, or the SOSA, represents an original alternative to HTS. It consists of two steps. Screen newly identified pharmacological targets of a limited set, approximately 1,000 compounds of well-known drug molecules for which bioavailability and toxicity studies have already been performed and which have proven usefulness in human therapy. By definition, in using such a library, all hits that are found are drug-like. Number 2, optimize hits by means of traditional, parallel, or combinatorial chemistry in order to increase the affinity for the new target and decrease the affinity for the other targets. The objective is to prepare analogues of the hit molecule in order to transform the observed site activity into the main effect and to reduce strongly or abolish the initial pharmacological activity. The rationale behind the SOSA approach lies in the fact that in addition to their main activity, almost all drugs used in human therapy show one or several side effects. An example is the antidepressant minoprine. In addition to reinforcing serotoninergic and dopaminergic transmission, this aminopyridazine possesses weak affinity for muscarinic M1 receptors. Now we come to the third strategy, that is exploitation of biological information. A major contribution to the discovery of new active principles comes from the exploitation of biological information. This means information which relates to a given biological effect provoked by some substances in humans, in animals, or even in plants or bacteria. Exploitation of observations made in humans. The activity of exogenous chemical substances on the human organism can be observed under various circumstances. Ethnopharmacology, popular medicines, clinical observation of secondary effects or adverse events, fortuitous observation of activities of industrial chemical products, etc. Because the harvested information is observed directly in humans, this approach presents a notable advantage. Now we come to another category, that is new uses for old drugs. In some cases, a new clinical activity observed for an old drug is sufficiently potent and interesting to justify the immediate use of the drug in the new indication. Amiodarone, for example, was introduced as a coronary dilator for angina. Concern about corneal deposits, discoloration of skin exposed to sunlight, and thyroid disorders led to the withdrawal of the drug in 1967. However, in 1974, it was discovered that amiodarone was highly effective in the treatment of a rare type of erythema known as the Wolff-Parkinson-White syndrome. Accordingly, amiodarone was reintroduced specifically for that purpose. Another category is the fortuitous discovery of activities of industrial chemical products. During the industrial manufacture of nitroglycerine, toxic manifestations due to this compound, particularly strong vasodilating properties, were observed in workers. From there came the utilization of this substance and later on of other nitric esters of aliphatic alcohols in angina pectoris and as cerebral vasodilators. Next, exploitation of observations made in animals. We find here all the research done by physiologists that has been the basis of the discovery of vitamins, hormones, and neurotransmitters and the fallouts of various pharmacological studies when they were performed in vivo. Other observations made on animals, often in a more or less fortuitous manner, have led to useful discoveries. An example is provided by the dicumarol-derived anticoagulants. Another type is exploitation of observations made in the plant kingdom and in microbiology. A particularly rich contribution of this approach in the therapeutic area has been the discovery and the development of penicillin. It initiated the discovery of many other major antibiotics such as chloramphenicol, streptomycin, tetracyclines, and rifampicin. In conclusion, whatever its origin may have been, the use of biological information constitutes a rich source of new lead molecules for research. Now, fourth strategy is planned research and rational approaches. The approaches that we have described up to now owe a great deal to chance or they lack originality. A more scientific approach is based on the knowledge of the incriminated molecular target, enzyme, receptor, ion channel, signaling protein, transport protein, or DNA. Now, let's talk about L-DOPA and Parkinsonism. A historical example in which the key information that rendered possible a rational approach to drug design is the discovery of the usefulness of L,3,4-dihydroxyphenylalaniline, L-DOPA, in the treatment of Parkinson's disease. Thus, since it was observed in patients suffering from Parkinsonism that the dopamine levels in the basal ganglions were much lower than those found in the brains of healthy persons, a symptomatic but rational therapy became possible. This therapy consists of administering to patients the L-DOPA. This amino acid is able to cross the blood-brain barrier and is then decarboxylated into dopamine by brain DOPA decarboxylase. 95% of the DOPA administered by the oral route is decarboxylated in the periphery before having crossed the blood-brain barrier. To preserve the peripheral DOPA from this unwanted precocious degradation, a peripheral inhibitor of DOPA decarboxylase is usually added to the treatment. An additional improvement of the treatment is the simultaneous addition of an inhibitor of catechol or methyltransferase such as tolcapone or entacapone. Fifth strategy, that is applying biophysical technologies and computational methods. This last strategy combines the increasing knowledge that we have on various targets and the new technologies that we can apply to assist the discovery of small molecules interacting both physically and functionally with the target protein. Progress in molecular and structural biology technologies allowed the identification and experimental characterization of several hundred molecular targets and facilitated the design of drugs at a more rational level. First is biophysical technologies. Knowledge of the three-dimensional structure of protein targets has the potential to greatly accelerate drug discovery. But technical challenges and time constraints have traditionally limited its use to lead optimization. Large panels of biophysical methods are currently used, such as protein crystallization, ligand-protein co-crystallization, and soaking, ligand-protein H3 and N15 NMR, surface plasmon resonance, differential scanning, fluorimetry, and mass spectrometry. Now, let's understand how hits are generated from fragments. This approach starts from the screening of a fragment set by technologies allowing the measure of low affinity interaction with the target protein. The protein characteristics determine the most appropriate technology to be used. For example, in general, it is difficult to perform NMR with proteins larger than 14 kilodalton. However, crystallography could provide valuable structural information. As the size of the compound does not usually exceed a few thousand, selection criteria tailored to the target should be established. Following hit identification, the challenge is in the conversion of a fragment having a binding affinity in the millimolar range into a compound showing a biochemical effect. The crucial step is the growing of the ligand toward higher binding and biochemical activity on the target using the structural information gained from NMR or crystallography. Next, finding extra interaction, the example of a covalent drug. Covalent drugs are not new, and chemists did not wait for these technologies to design such products. Today, however, structural biology information helps to make the design of such molecules more efficient. This method can be applied to the generation of covalent inhibitors, taking advantage of the identification of a nucleophilic group. The acceptor functionality can be modulated according to the expected profile of the molecule, which is carefully evaluated by specific biochemical assays. The anti-cancer drug ibrutinib derives from this structure-based approach. Now, let's come to computational methods. First, virtual screening. Virtual screening can be defined as a set of computational methods that analyzes large databases or collections of compounds in order to identify potential hit candidates. This search can be performed on corporate libraries and/or on virtual libraries. These in silico experiments can complement HTS. They can also be performed prior to experimental screening or after HTS to rescue some compounds potentially missed by the in vitro readouts, so-called latent hits. An example of molecule that has been developed with the help of in silico approaches includes tyrofibran. Tyrofibran is a reversible antagonist of fibrinogen binding to glycoprotein 2B or 3A that prevents blood clotting. This compound is one of the first FDA-approved drug whose origins can be traced back to a pharmacophore-based virtual screening design. Next, scaffold hopping. Scaffold hopping aims to discover structurally novel compounds by replacing the central core, also called scaffold, chemotype, or framework, structure of a known active compound. The principle of scaffold hopping is to retain enough similarity in the template compound to keep the activity while altering the central core and changing the properties of the molecule or moving it into the novel chemical space. Numerous methods that enable the generation of scaffold hopping exist and have been compared, including the heterocyclic replacement, ring opening and ring closure, peptidomimetics, topology-based hopping, 2D fingerprint, and 3D pharmacophore. That's all for today's lecture. Hope you have understood it well. Thank you and goodbye.